Hello everyone. This will be the recorded discussion of our lecture regarding the proteins. We are already done with the two bioinorganic substances. So what are again the bioinorganic substances? We have here the carbohydrates, lipids, protein, and nucleic acid. Okay, so we are already done with the carbohydrates, lipids, and protein. We are on our third bioinorganic substance, which we will be focusing about the protein. First, let us know what is a protein. Now, protein is a naturally occurring and branched polymer in which the monomer units are amino acids. So, protein, why is it important that we study protein? Next to the water, we know that water is one of the most abundant molecules in the cell. Next to the water is the protein, which accounts to 15% of overall cells of the cell's overall mass. So 15% of cells overall mass. The elemental composition of proteins are carbon, hydrogen, nitrogen, and oxygen. Okay, so the average nitrogen of protein content is about 15.4% mass. Okay, please make sure to take down notes as some of this part of our lecture would not be found in either of the PowerPoints or the handouts provided. So again, please make sure to take down some of the notes. Okay, so let us continue. What are proteins needed for? So proteins are needed for synthesis of enzymes and other certain hormones. It is also needed for synthesis of some other blood components. It is needed for the maintenance and repair of existing tissue and synthesis of new tissue. So the human cells contains about 9,000 different kinds of proteins, while the human body contains 100,000 different kinds of protein. Okay, so please take note that there are special proteins, no? So these special proteins might contain a phosphorus and iron. So for example, you have here casein. So this is the main protein of the milk. So this casein, it contains phosphorus. So for what is the importance of phosphorus? So the importance of phosphorus, it is needed for the diet of infants and children. Another example we have is the hemoglobin, which is an important oxygen transporting protein of the blood, which contains iron. So we know that proteins are the monomer units of amino acid. Now what are the amino acid? As we all know, or as we are all familiar with, amino acids are the building block of protein. Okay, so these are the molecules that combine to form protein. Now each amino acid contains a carbon group, an amino group, carboxyl group, hydrogen, and the R group. So the R group that is present in the amino acid is also called the amino acid side chain. So the R group is very important because it differentiates on what type of amino acid is present. Side chain varies differently in sizes, shape, charge, acidity, functional group present, hydrogen bonding ability, and chemical reactivity. Okay, now let's look at this example. Take note that all of the amino acid contains an amino group. It contains a carboxyl group and a carbon atom. So if the R chain contains only hydrogen, this is an example of glycine. So the part that differentiates your amino acid, again, it is the R group, or it is also known as the amino acid side chain. We have here another example. If the side chain contains a methyl group, we call this the alanine. So 8 out of 20 standard amino acids are nonpolar. What do we mean when we say nonpolar? These are hydrophobic or they are insoluble in water. Okay, so usually our groups are nonpolar amino acid. Okay, so when present in protein, they are located in the interior of protein where there is no polarity. Now these are the polar amino acids. So there are 3 types of polar amino acid. These are found in your handouts. So we have polar neutral, polar acidic, and polar basic. When we say polar neutral, it contains polar but in neutral side chains. There are 7 amino acid which belongs to this category. Next, the polar acidic. It contains carboxyl group as a part of their side chain. There are 2 amino acid which belongs to this category. Lastly, the polar basic. Polar basic contains amino acid group as part of their side chain. There are 2 also amino acid belongs to this group. Now here are some of the examples of the nonpolar amino acid and the polar amino acid. For the nonpolar, we have here the alanine, cysteine, glycine, isoleucine, leucine, methionine, phenylalanine, proline, tryptophan, tyrosine, and valine. For the polar amino acid, arginine, asparginine, aspartic acid, glutamine, glutamic acid, histidine, lysine, serine, and threonine. Now, as we are keep on discussing the nonpolar and polar, ano ba talaga yung ibig sabihin ng nonpolar and polar? Let us now differentiate polar versus nonpolar amino acids. So, polar amino acid actually means that they are oppositely charged, while the nonpolar amino acids are equally charged amino acids. So, both polar and nonpolar amino acids are a type of covalent bonds. Okay, so the negative charge from the electron is not evenly distributed in the molecule, which causes a dipole mole. So what is a dipole mole? So a dipole moment occurs when one end of the bond is positive and the other is negative. So we have here, for example, of polar bond is the bond between the hydrogen and oxygen. So the bond is classified as polar because it has a large electronegativity difference. So the electrons in the hydrogen are more attracted to the electrons in the oxygen because oxygen is negative, electronegative. So it does not share an equally distributed electrical charge. While the nonpolar amino acids are equally charged. Okay, so an example of a nonpolar amino acid we have here is chlorine. So chlorine contains 2 chlorine atoms which shares equally because the electronegative difference between 2 atoms is 0. Let us now discuss some of the amino acid. First we have here is the cysteine. So cysteine is said to be unique among the other standard amino acid because it contains a sulfidyl group. Okay, so again, cysteine is unique because it contains a sulfidyl group. So among the standard amino acid, dalawa lang ang nagko-contain ng sulfidyl group and one of this is the cysteine. Okay, so cysteine is known to be a non-essential amino acid because our body can create its own cysteine. Okay, so it is important. It is an important protein in the nails, skin, and hair. It is also important for making collagen. So next are the peptide. Peptide is a short chain of amino acid. Under proper conditions, amino acid can bond together to produce an unbranched chain of amino acid. So peptides are known to help for building muscle, boost weight, and also for muscle recovery. So the amino acid in a peptides are connected to one another in a sequence by a bond called the peptide bonds. So the covalent bond between amino acids are called the peptide bonds. So the bond between two amino acids are called dipeptide bonds. The bonds between 10 to 20 amino acids are oligopeptide bonds. And bonds between larger number of amino acids are the polypeptide bonds. So the peptide that contains the same amino acid but present in different order are different molecules with different properties. So these are known as the isomeric peptides. Next, let us now discuss some of the biochemically important peptides. Let us now discuss the biochemically important peptides. So each of these biochemically important peptides contributes to different function, like the hormonal action, neurotransmission, and antioxidant activity. We have here first the small peptide hormone. There are two best-known small peptide hormone is the oxytocin and vasopressin, which are both produced by the pituitary gland. Oxytocin and vasopressin are known to be nonapeptides. When we say nonapeptides, these are 9 amino acid residues. So these contain 6 of the residues held in the form of a loop by a disulfide bond formed between two cysteine residues. Some of the important function of these two, for vasopressin, it is known to regulate lactation and water balance. They are also a potent regulators of skeletal integrity. While oxytocin is known to be important to stimulate uterine contraction in labor and childbirth. It also stimulates contractions of breast tissue to aid in the lactation after birth. So next is a small peptide neurotransmitter. Enkephalins or peptide neurotransmitter which are produced by the brain and bind receptor within the brain. So enkephalins help reduce pain. There are two best known enkephalins. So these are the met enkephalin and the Lu enkephalins. Next, we have the small peptide antioxidant, which is the glutathione. Glutathione is famously known as a whitening product, but its main function is an antioxidant. It is a regulator of oxidation. Glutathione is an antioxidant and protects cellular contents from oxidizing agents such as peroxides and superoxides. Let us now move on to the general structural characteristics of protein. This part of the discussion is found in your handouts. You can listen this discussion or this recorded lecture by following the notes in your handouts to make it more easier and clearly understand the handouts. So the general structure characteristic of proteins. Earlier we have discussed we have defined its general definition. It is said that the protein is a naturally occurring unbranched polymer in which the monomer units are amino acids. Again, amino acids are the building blocks of protein, meaning that proteins are made up of several amino acids. So a specific definition is that a protein is a peptide in which at least 40 amino acid residues are present. The term polypeptides and proteins are often used interchangeably used to describe a protein. So there are several protein which contains greater than 10,000 amino acid residues. Common protein contains 400 to 500 amino acid residues. Small protein contains 40 to 100 amino acids. And more than one peptide chain, take note, may be present in a protein. You can post this recorded lecture anytime so you can get your notes and follow the discussion, again, for easier and clear understanding. Okay, so when we say monomeric, these are monomeric protein which contains one peptide chain. Multimeric, these are multimeric protein which contains more than one peptide chain. So now, classification based on chemical composition. So only amino acid residues are present. These are the simple protein. As found in your handouts, these are the simple protein. Next classification is one or more non-amino acid entities are present in its structure. These are the conjugated protein. One or more polypeptide chain may be present. A non-amino acid component may be organic or inorganic. So lipoprotein, these are the protein which contains lipid prosthetic groups. Glycoprotein contains carbohydrate groups. Metalloprotein contains a specific metal as prosthetic groups. So we will now move on to the four types of protein or the structure of the protein. So there are four types of proteins. So we have the primary structure, the secondary structure, tertiary structure, and quaternary structure. So again, these are all found in your handouts. The primary structure of protein, this refers to the order in which amino acids are linked together in a protein. The secondary structure is the arrangement of atoms of backbone in the space. So there are two most common types. We have the alpha helix and the beta pleated sheets. The tertiary structure, this is the overall three-dimensional shape of proteins. And the quaternary structure, these are the protein which refers to the organization among the various peptide chains in a multimeric protein. So I will be uploading a supplemental video, an animated video wherein you can visualize and understand the structure of the protein clearly. Before moving on to our next topic, please take a pause and watch the supplemental video on the four types of protein which are uploaded in your canvas. I have uploaded a video to help you understand and visualize the structure of a protein clearly. Now that we have understood the structures of protein, hopefully that you have watched the video presentation, we will now move on to the classification which are based on the shape. So there are three types of proteins that are based on shape. We have the fibrous protein, globular protein, and membrane protein. So the fibrous protein, these are protein molecules with elongated shape. Fibrous protein are generally insoluble in water. They tend to have a simple, regular, and linear structures. While the globular proteins are protein molecules with peptide chains folded into spherical or globular shape. Globular proteins are generally water-soluble. So they are hydrophobic amino acid residue in the protein core. Lastly, the membrane protein, these are associated with cell membranes. They are known to be insoluble in water, and they help in transport of molecules across the membrane. So we have two examples of fibrous protein. We have the alpha-keratin and the collagen. The alpha-keratin provides a protective coating for organ. Major protein constituents of hair, feather, nails, horns, and turtle shells. mainly made of hydrophobic amino acid residue. The fibrous protein collagen is the most abundant protein in humans. So we have about 30% total of body protein. So collagen is the major structural material in tendons, ligaments, blood vessels, and also skin. It is also an organic component of bones and teeth. So the collagen is rich in proline, which is up to 20%, and it is important to maintain the structure. Next, we have our globular proteins. An example of globular proteins we have here is myoglobin and hemoglobin. So myoglobin is an oxygen storage molecule in muscle. Oxygen stored in myoglobin molecule serves as a reserve oxygen source for working muscle. It has a higher affinity for oxygen than hemoglobin. Hemoglobin, on the other hand, is an oxygen carrier molecule in blood. It transports oxygen from lungs to tissue. It can transport up to 4 oxygen molecules at a time. Iron atom in heme interacts with the oxygen. Next is the membrane protein. So the membrane protein actually covers a whole lot of new lectures. No? So just to give you its function, so the membrane proteins function in the junction, enzyme, transport, and recognition. So for junction, this serve to connect and join two cells together. The enzymes, these are the fixing to membranes localizes metabolic pathway. The transport is responsible for facilitated diffusion and active transport, and the recognition may function as marker to cellular identification. Some of the other proteins are found in the handouts I have uploaded in your Canvas. Please read some of these proteins and familiarize yourself with each of the terminologies. You don't have to memorize because our exams will be multiple choice. Please make sure that you have read the handouts. Some of the other important proteins are found in our handouts. So that is the whole coverage of our protein recorded lecture. I hope you have understood. Again, as a reminder, I am telling you this repeatedly. Please, if you have questions or clarification regarding the recorded lecture, don't hesitate to send a message and ask questions. Thank you for listening.